All right, everybody, welcome back. We are continuing on to Module 6.2, where we're going to specifically look at bacteria. So again, we've been talking about prokaryotic diversity, and that was the subject of our last lecture, and really understanding what makes prokaryotic organisms unique. And now what we're going to be doing is diving in specifically to the domain bacteria and what makes bacteria a unique prokaryote. So I wanted to start this off with a study that actually I was involved in during my graduate work, working with Dr. Hagen, and looking at a bacteria that is common in urban favelas in Brazil. And it's a bacteria that is a symbiote with rats. And when the rats shed it in their urine, it can survive in soil and water. And then humans can become accidental hosts and become infected when you walk through this water or mud, and you can actually pick up the disease, and it can be really quite serious. So, we probably discussed quite a bit about other epidemiology studies looking at bacteria, but I thought that since we were going to be talking specifically about bacteria today, I would highlight a bacteria that is a neglected bacteria that's occurring in a lot of urban environments with lower levels of infrastructure. So again, I want to sort of start this lecture by centering us where we're at in the tree of life, if you will. So we're in the area of prokaryotes, which include archaea and bacteria, and specifically in bacteria. And today we're going to be sort of talking about a wide spectrum of bacteria, looking at things that live symbiotically, things that are pathogenic, fermenters, the motility of different bacteria. And all of these are ultimately what start to form these different clades within the domain bacteria. So we're going to talk specifically about different characteristics that make up a bacteria and ultimately what makes them unique. So again, first I wanted to dive into the structure of bacteria. What characteristics do all bacteria have that sort of make it unique? And the first thing is a cell wall. Now there are a couple exceptions to this, but the vast majority have this cell wall, and that is very much unique to bacteria. They also have tremendous genetic diversity. This picture here is actually something that you've probably heard of as part of the gut microbiome. And this is actually a zoomed in picture of bacteria that colonize the intestines of human health. And it's a huge area of study right now for looking at the effects of microbiome in human health. Now, bacteria, there can be a lot of differences in nutrition, metabolism, lifestyle, and structure. And that, again, is what is ultimately going to differentiate bacteria into those different clades in the tree of life. And there are a couple different broad types of bacteria. The first one's being autotrophs. So basically organisms that can make their own food from material in a non-living environment. Auto meaning self, troph meaning to eat. And then we have heterotrophs, which have to rely on other organisms as a food source. So we're going to dive into both of these specific broad branches of bacteria a little bit later on in the lecture. So in terms of structure, there are three main branches of bacterial structure. The first being spherical. And a good example of this is our old friend Staphylococcus aureus. So a way that you can determine whether or not a bacteria has a spherical structure is they'll have the word cocci is somewhere in their name usually. So Staphylococcus aureus tells us that it is a spherical bacteria. Likewise with rod shape, but the root there is bacilli. So for example lactobacillus. Again, understanding rod-shaped. And lastly, we have spirochetes, which come in a spiral shape. So for example, the bacteria that we were looking at the beginning of the lecture as our example, leptospirosis, again, looking at that spherical type structure. Oh, goodness, sorry. Cat just jumped on top of me. So continuing looking at bacterial structure, one of the things that is unique is this capsule. So this is a sticky coating that surrounds the cells and helps protect them. And ultimately, this capsule is one of the things that really helps bacteria to survive in such varied environments. Another structure that's really unique to bacteria is a flagella. Now, again, not all bacteria will have this, but it's essentially a tiny whip-like structure that helps project the cell and, again, to help it move. And again, thinking of what these structures actually help these organisms to do. So this actually is going to help bacteria become motile and find the best environment for it to survive. Another appendage that is helpful for both adherence and also motility is pili, which are these tiny hair-like structures that can form on the outside of bacteria. So now that we've sort of examined the broad structures that really help make bacteria be bacteria, let's dive into sort of these larger clades. So the first one being autotrophs. And again, these are bacteria that are making their own food and nutrients. And by far the most abundant and probably the most important autotroph is cyanobacteria. It's the largest group. It's found in oceans, fresh water, exposed to rocks and soil. And what's really unique about this organism is that it photosynthesizes. And cyanobacteria are indeed the oldest photosynthesizers on the planet. And what's interesting, particularly about these cyanobacteria, is it's hypothesized that the first plant cells actually engulfed cyanobacteria, and that's how they were able to start photosynthesizing. So what about bacteria that need to get their nutrients from other sources? Again, there's sort of broad clades that exist within them. So the first one that we're probably most familiar with are decomposing bacteria. So they recycle trapped carbon and other elements. So these are the bacteria that make compost heaps work and compose into healthy soil. Another sort of lesser known are bacteria that we actually use for bioremediation. So there's actually been a very recent breakthrough looking at bacteria that actually will ingest plastic and recycle, and ultimately could potentially have huge roles in recycling. There's also likewise bacteria that have been employed in oil spills that are able to eat and synthesize oil. And lastly, we have another group of fermenters. And mostly we think about fermenters, we think about bacteria that are important in food production. So we think about yogurt, we think about cheese. These are all examples of bacteria, or bacteria specifically heterotrophs, that are important to the human food supply. Another type of heterotroph are those that live in symbiosis with other organisms. For example, there is a famous bacteria called Wolbachia that, when it's introduced into wild mosquito populations, actually decrease the amount of dengue virus that can be transmitted by mosquitoes. Dengue being a very important tropical disease passed on by mosquitoes. So there's actually studies that are looking to essentially inject this Wolbachia strain into wild mosquitoes and then have these mosquitoes breed with this gut flora, and potentially using that as a way to decrease disease throughout um, Latin America and um, Southeast Asia. There's also like potential beneficial um, bacteria, such as those that bioluminesce within squids. So you can see that that again, these bacteria are living in symbiotic relationship with the squid and providing it displays for mating and communication. So probably the bacteria that we're most familiar with are those that are pathogen-causing agents, and they cause disease by producing toxins that become harmful to their host. So here we have those three examples from earlier. We have rod-shaped bacteria, E. coli. We have Staphylococcus aureus, and we have leptospirosis. But again, these don't are actually are don't cause a lot of the biodiversity that we see within the domain bacteria. So just keep that in mind, that bacteria are much more than just potential pathogens. They have different roles and are heavily abundant in most ecosystems. Okay, we're going to pick up next time going in and examining archaea. All right, see you soon.